Good morning. Thank you for your coming. I am glad to introduce you Professor Gabrijel Ondrasek from the University of Zagreb. He is the head of Department of Soil Amelioration and is our guest in the Fondecyt project. Today he will introduce us the seminar entitled Trace Element and Organic Interaction in Food Safety and Environment Protection. Professor Ondrasek is author of over 30 papers, scientific papers. Also is the author of 2 books and participate as author or co-author in over 13 academic books, scientific books, in collaboration with other researchers. Also, he is the coordinator of different projects in Europe and Australia. Also, these projects are related with the water management, and his research line are the management of water and salinity in soils, and also involved in agroecosystems. Professor Ondrasek, I am glad to introduce you.
Thank you, Cristian. Thank you. Okay, ladies and gentlemen, my greetings and greetings from my home institution. But before I start, I would just like to say a few things. I would like to express my great thanks to Dr. Cristian Gergichevich, to my dear and friend and my colleague from Chile, and also I'm very grateful to Alex Ziguel, and also I'm very grateful to director of BioRen Center, Dr. Maria de la Luz Mura. Before we start, also I would just like to introduce you, because this is my first out of 3 presentations, just short introduction with my home country and my home university. It's actually, as you can see, a very small country. The youngest and the newest actually country in European Union. Very small in population. If you compare it with Chile, then you can get better sight about the area, about the GDP per capita. They are similar, but it's much smaller in other parameters than Chile. But we are maybe the most famous that you might know according to sport activities.
Yes.
We are proud of our sport activities because we are actually great sport nation. Not only in football, but it is the recent activity that we succeeded very well. But also we are a really great nation in other individual or team sports, and really we are proud of it. So, few things about my university. It's actually very old, established in 17th century. Has, as you can see, 30 faculties and 3 art academies. Many of students, over 70 hundreds, lots of teachers, and so on. So out of those 30 faculties, one is mine, Faculty of Agriculture, and it has 28 departments. One of those is my department of soil amelioration, and actually it's a great faculty related to agricultural sciences. There is over 400 of staff members. Alumni has over 12,000 of different students, mostly undergraduate, graduate, and PhD. We got 7 experimental stations on the field, in protected environment, and also as faculty we participate, or we did participate in different EU projects and with other countries. Also, I think it is good to know how we can improve our collaboration in between of Croatia, in between of my university and your institution, your university, BioREN, and so on. Maybe you might don't know all of you, but some knows. Actually, currently it is possible to cooperate among different projects, calls founded by European Union. For instance, Horizon 2020 is actually one of those, which is the biggest EU research and innovation program, which supports different research activities among countries, institutions, in different at different continents, underpins collaboration between multidisciplinary science and industry, and so on and so on. There are really huge and dispersed possibility to take some of collaboration within this Horizon 2020 program. So I would suggest you that you just take some time and search on the website and to see what are possibilities for you, because it's really lots of information. Also, on the field of possible collaboration in academic teaching, training, there is also one program called Erasmus+, which supports education, trainings in Europe, in between of European Union countries, but also in between of other countries. For instance, Karina is coming within this program next year. Some other people will come to Croatia from Chile within this program. Also, some people will come to Chile over this program. This is also one of possibility to improve our collaboration in this field, so teaching and training. Also, I am very proud as a program coordinator of one master study program in English language. Actually, this master study program, called under the acronym Interenagro, is very young study program in English related to environment, agriculture, and resource management. So we have run it study a few years ago, and currently we have students from USA, from Czech Republic, from Philippines, and so on. So it is still young, but we expect that in the future we will attract more and more students, maybe from Chile. We would like to have some Chilean students on this study, and Karina will take some courses from its curriculum. As you can see, it lasts 2 years, actually 4 semesters, with different obligate and elective courses. So that would be just few things from my country, from my university, and now we are moving on actually today's presentation, which is going to be about trace elements and organic and inorganics interactions related to food safety and environment protections. So just few things about topics background, which is actually quite complex, I have to say. each subtopic is quite complex by itself, and in combination with these three subtopics, it makes the situation even more complicated. So, metal actually, or trace element, contamination, it's actually acronyms. A few minutes ago, I was speaking to one colleague about this. Actually, every country has problem with metal contamination, with trace elements, and so on. And you will see why are they so important from the perspective of food protection and environment protection, because you will see only few trace metal elements or trace elements are essential, and for us, for plants, for animals, but in very, very small concentration, in small amounts, which can be very easily exceeded, and after that they express harmful effects to us. Another problem also present around the world is salinization, salinization processes in land areas, in soil, but also in water resources, which also affects food production, reducing the yields, but also combination of metals, of certain metals and salinity. You will see that also can impact and quality of food that we eat. And the last one, but not the less important, is actually soil organic matter. In what respect? In respect to its decline, to its depletion in agri-ecosystems. You will see organic matter is highly complex soil parameter, and it's really important from this perspective, from food protections, from metals and some other pollutants, and especially some substances of soil organic matter. You will see how can they affect metals mobility and bioavailability in different pedosphere metrics. So let's go step by step, subtopic by subtopic. As I said, soil organic matter is one of the most complex, but also and most varying and functional pedosphere variable. For instance, in soil-affected areas, like sandy areas in semi-arid and arid regions, its common content is less than 0. something%. But contrary, in humid region, it's dominant. It dominates as soil phase. For instance, in organic histosoils from uplands of humid and boreal climate zones on northern hemisphere, it can yield over 100% of soil content. Also, soil organic matter has very important roles in cycling of some nutrients. At first, in cycling of carbon, but also in nitrogen and some other elements, and also in some functions of soil as one multiplex ecosystems. So, in cycling, soil organic matter has a very important function in these processes. So, in decomposition, mineralization of some organics, after that in their polymerization, then emission of some nutrients at first of carbon and some other nutrients. And opposite in functions in different soil functions, organic matter is very important from biological perspectives because it serves as energy and as nutrient source. It improves biodiversity, health of soil, and so on. Also, from chemical perspectives, it's important in carbon and nitrogen sequestration, in absorption of different organics and inorganics, you will see, and thus can prevent their leaching to groundwaters, to surface waters, and actually it enhance cation exchange capacity because of huge reactive interface. Also, from the physical properties of soil as matrix, organic matter in soil stabilizes soil structure. It aggregates the soil particles, prevents soil compaction, improves also soil water capacity, which is very important from my perspective, because actually my department in the past was actually involved in water management in agri ecosystem, and actually this was very important for me at the beginning. But also, and from some another perspective, for instance, for agroeconomists, it is also very important from that perspective because it serves as an energy source, also reduce some production inputs, for instance, in organic agriculture. Over the organic fertilization, you don't need so many chemical fertilizers, okay? And of course improves plant crop yield and quality, so you can obtain maybe better and higher price for your organic products. So that's only short and summarized presentation of organic matter relations in cycling and functions inside of soil pool. As I said, in European Union, we got lots of problems with organic matter depletion. Almost half of our soils, actually topsoils, up to 30 centimeters, are very low or low in organic matter content. So why is that situation? Because of intensive activities in agri-ecosystems. Also, another problem is that almost half of our soils have medium, which is also not good content for organic matter. And especially that problem is visible in Southeastern Europe, in Mediterranean countries like Spain, Italy, also Croatia, Greece, Albania, and so on. You see that 74% roughly in topsoils up to 30 centimeters are very low in organic matter content. It should be much higher. So, and according to some recent, let's say, studies, estimated costs caused by organic matter declining are estimated even up to over 7 billion annually, but that costs don't include actually and soil the damage caused to ecological functions of soil, because you can't just express that value. It's difficult to express in money such value. Also, my today's second part of presentation will be specifically related to specific soil organic matter pool. So, if we are talking about different organic matter pool, then today we won't talk about living biota, which is important, of course, but we won't talk today about that. Then we will talk about specific part of non-living organic matter, actually about this organic pool, about humics in the pool and their importance in food safety and security. You can see that actually that pool, which means HMW, high molecular weight organics, dominates actually in the solid organic matter pool. Also, very important, organic matter pool is actually potentially dissolved organic matter pool, which is also important. You will see especially at the rhizosphere scale. So let's move further. Today also I won't talk much about the role of soil organic matter in cycling of certain nutrients, in certain elements like is carbon, but you have to be aware that actually organic matter has really great role in that actually cycling of certain nutrients, like is for instance carbon dynamics and cycling, especially on the global scale. Soil organic matter has really important role is in that also process. But because of let's say duration of my presentation, we won't much talk about that. Hopefully, Cristian and me, we are hope that you will soon can read about that in, I hope so, in soon published paper. Yeah. Okay. And let's move for a second subtopic, actually trace elements or metal elements, I like this term trace elements. Trace elements. It's maybe more modern than but also you can find another expression for that term, like heavy metals, like metalloids and so on. Actually, I'll talk about trace elements. So it's actually relatively large group of essential and non-essential elements. Some are very toxic for us, for living organism, but in normal non-contaminated soils, such elements are actually present in trace amounts, and because of that they are called trace elements. But on contaminated sites, you will see they are really they can be present in much higher concentration. For instance, several folds higher than in normal non-contaminated soils. Also in European Union, contamination by certain trace elements is recognized as also a huge problem, and one of the main threats to our environmental resources and to our health. For instance, according to recent studies, it is estimated that in European countries is over than 3.5 million sites that can be considered as contaminated, so with elevated concentration of certain one or more elements. While out of that, over 500,000 of such sites, 500,000 are considered as really contaminated. Also, the cost, as you can see, caused by metal contamination is really high, even higher than previous one. Can be up to 70 billion euros annually. And also this value not includes damage to ecological value of certain site, because you can't actually express that in money so easily. Also, as I said to colleague before on, sorry, I forgot your name. Francisco, yeah. We do have in Croatia also such, let's say, hotspots with metal elements. For instance, this is a situation of Croatia. So, Italy, Hungary, Austria, yeah, and this is actually Adriatic Sea. Very small Adriatic Sea, connected to Mediterranean Sea. And you can see some examples of hotspots related to excessive concentration of lead and zinc. Where are those hotspots? For instance, in this northern region, which is in the neighborhood with Slovenia and Hungary, this is Serbia, you can clearly see from recent study, really recent study, Halamić and Minko 2009, confirm hotspots not only for these two metal elements, but for and some others, but I will show just two these examples. Really nice hotspots. Actually, these hotspots are in very close relations. Actually, they are detected along the Drava and the Drava River and the Danube River on the border. Croatia don't have such intensive industrial activities, so we explain these hotspots as a cause of fluid deposition from the upstream regions, industrial regions, because Drava and the Danube River were transferred probably those metal elements from upstream industrial regions, and over the time those metals were deposited by so-called fluid deposition around the courses of those two rivers. So you can see in the topsoils of and also these hotspots are in they are on agricultural soils. Those soils are very fertile, with very good physical conditions. It's a fluid soils with very good water relations in their soil profile. So here agricultural activities are very intensive, and you can see that around these hotspots actually is very present lead and cadmium. Over 1 gram per kilogram of dry soil, and here over 600 milligrams per kilogram of soil, which is several folds higher than normal topsoils. Also, another problem, salinization process present in soils or in water resources, as you can see, is also a very big global problem present at every continent. Of course, and in your continent, I don't know the specific data for Chile, but for instance in Croatia we do not have such big problem with salinization. We got about several thousand hectares of saline soils, let's say, and they are mostly located here along the Adriatic coast in arable and very fertile soils developed along some river estuaries, which are connected to Adriatic Sea. You will see later on. For instance, one of those delta is Neretva River Delta. Then Vrana Bazen, and so on and so on. But in general, Croatia don't have such big problem so far with salinity in soils. But who knows? Under the global climate change, with higher temperature, more intensive evapotranspirations, and so on, that problem can be even bigger, not only in my country but also in other countries. And as you can see, it is present in almost all countries globally, and affects 1 million. Yeah, that's right. And that problem is also especially present on areas in agri-ecosystems that are irrigated. That will be my second and third talk next week. So that problem salinity is really specific for irrigated area, and unfortunately it has annual increase. You see significant increase per year, every year at global scale, which also can be accelerated under the current global warming and so on. Okay, I'll show you now, after this brief introduction to each subtopic, I will show you some studies related to metals interaction and their implications to food security. For instance, you will see some recent exams related to zinc and cadmium about that. So, as you know, unfortunately, plants have very big ability for selective extraction of essential elements only. So they also, beside of essential, take up and beneficial, but also and non-essential elements that can be very toxic, carcinogenic, teratogenic, and so on. Unfortunately, yeah. So, because of that, you have to know that some of the most dominant parameters that control trace elements chemistry in the soil and their mobility and availability to plants are pH and organic matter content, soil organic matter content. For instance, you will see here the influence of pH and dissolved organic matter concentration on cadmium speciation. For instance, if we got in the solution relatively small concentration of cadmium, 10 micromoles, and relatively small concentration of dissolved organic carbon, you can see over the pH range how those two species, one is free cadmium species and another is species of cadmium complex to organic matter, how they react. For instance, in the pH range from 4 to 7, free mobile cadmium dropped by more than 60%, while in the same time organocomplex cadmium 4 increased by 7-fold. So it is important to know that, because actually this pH range is quite relevant for agri ecosystems. For instance, in this experiment you will see how is one very common crop, like is radish, how is able to accumulate, to uptake some toxic and some essential elements in very, very short term of exposure. For instance, what we have done. We have grown in fully controlled environment, in growth chamber. We have grown radish plants for 17 days in normal nutrient solution for radish plant. And after that, we were just for 24 hours expose the root, expose the radish nutrient solution to relatively small concentration of zinc 70 and cadmium treatment at two different concentration. For instance, one was high, another was low. High means that concentration of zinc 70 and cadmium was equimolar. It included 2.2 micromoles of zinc and cadmium, and in the other case, zinc was low and cadmium was the same as in previous one. So, what have we discovered? Okay, before that we have done the next. So we have taken the root apex, very small parts of root tips. You can see here, and prepare them that root tips for nanoSIMS analysis. You will see later on what is that. And all the rest root, fruit, and leaves, according to the same procedure, we were taken the small part of fruit, the small part of leaf, and exposed them to nanoSIMS analysis, and the rest of each anatomic part of radish was examined by ICP-MS analysis. So what was obtained? Yeah, just few things about nanoSIMS. Actually, this was done at the University of Western Australia with very, very expensive and state-of-the-art instrument. I won't bother you with the specific things, but actually it is actually a unique special microprobe that you can map certain elements, isotopes and so on in certain samples and detect them if you have lucky to detect them, of course, yeah. So what we have done, first step was to detect, is that sample adequate for nanoSIM analysis? I mean, did the test plant uptake enough concentration of your metal of interest that you can examine that sample by nanoSIMS, which is quite expensive, and it would be wasting of time and money if you don't have it. But it's interesting, for instance, here you can see the results of MS results related to concentration of zinc 70 in different radish tissue, in root, fruit, hypocotyl that we are eating, and shoot everything the green about the fruit. At certain level, root high means 2.2 micromoles of cadmium and zinc, and root low means 0.5 of zinc and 2.2 of cadmium. We were surprised that actually that radish plant, which is actually not considered as metallophyte, as metal tolerant plant species, can uptake and transport those isotopes and cadmium in such short term period, like it's 24 hours only. So we have detected relatively high concentration of all in all anatomic parts, but of course the mostly, as we were expected, in the root. Similar situation was with cadmium, but in much bigger concentration, especially in root tissue. So it was it was confirmation, let's say, that we can go further with further analysis. And we have succeeded in mapping of certain elements. You can see here of maps of zinc, of cadmium, and potassium, and so on. Okay, picture is not so clear, but what was discovered by SIMS mapping was discovered next. Mostly zinc and cadmium were accumulated mostly in this outer apical root region called root epidermis, and toward to the root center, their signal was decreased. So it means that both metals of interest were mostly deposited in the outer root region, and towards to the root stem, to the root central cylinder, their concentration, their presence was low. Also, we succeeded to map those elements in fruit, in sections of fruit, and also in leaves, which is amazing, yeah. So it means that although cadmium is not important to radish and to most of living organism, it can relatively easily to be taken up and deposited in fruits that we are using every day. So we have to be careful about the environment in which we are growing our plants. Now we will see some examples related to metals and organics interactions. Why are they so important in relations to food safety and security? For instance, one interesting example is also with radish. I like radish and to eat and to do with radish. We can discuss it later on, but actually radish is very grateful test culture. Why? I can leave it for later on. But in second example, for instance, you can see what can be with certain trace elements if at first you grow the radish, for example, mostly in normal, let's say, nutrient solution, and after that, only for 10 days, expose the nutrient solutions with next. So what we have done? We had, let's say, controlled treatment, which means only full nutrient solution for radish, plus 4.4 micromoles of cadmium. And after that, we had 4 another treatments with 15 milligrams of humic acids. As you might remember, I'll just remind you, but humic acid is actually very important pool of stable soil organic matter. Humics actually, it's difficult to define, but it assumes humic acid, fulvic acids, and so on and so on. So in this case, in this example, we have used purified humic acid at different concentration, at 15, 75, 150, and 225 milligrams per liter of humic acid. In the presence of 4.4 millimoles of micromoles, of cadmium. So what we have obtained? Interesting results recently published. For instance, here you can see how actually humic acids had the influence this was control treatment, and actually this was the highest treatment with 225 milligram of humic acids per liter. Actually, the conclusion was that actually humic acid as very important soil organic matter portion decreases the uptake and distribution of certain trace metal elements, but without negative impact on the growth of tested culture. For instance, you can see that growth of radish was similar without significant influence, but for instance, accumulation and distribution of cadmium, copper, zinc and manganese was totally different in between of control plant and the treatment with the highest level of humic acids. You can see that accumulation of all of those metals of interest was the lowest with the addition of humic acids. Also very interesting and is this parameter. Actually, you can see here what was happening with chemical speciation in the rhizosphere of test plant. For instance, here in control treatment, cadmium, zinc, and cadmium, copper, and zinc were mostly present as free metal forms. And those forms are the most bioavailable for plants. While, contrary, in this treatment with humic acid, they were mostly, especially copper, was complex with humic acid. It wasn't present as free, but as complex with organic humic acids. The similar with zinc and with cadmium. But some other people, some other researchers obtained an totally opposite result than we have. For instance, Anglo and other, for instance, obtained a positive effect with the addition of humic acids, but methodology, conditions, very different. We won't discuss it, but they, for instance, obtained opposite positive effect of humic acids, for instance, applied to soil, but on concentration of cadmium in shoots of tobacco. Also, Bandera and other and associates also obtained positive influence of application of humic acids to metal content in test plant species, actually in shoots and roots. So there are and some other influence, but that influence is also was significant in relation to uptake of trace metal elements like cadmium, copper, and iron. Okay, guys. I forgot to tell you, you can stop me at any time if you got anything to ask, to discuss. I thought to leave some time for after my presentation, but if you don't understanding or if I'm not so clear, please just stop me and we can discuss that immediately. And now I will show you, let's say, the most complex triple interaction: organics, metals, and inorganic salts interaction in food safety and environment protection. I'll show you briefly the results of 4 experiments in controlled conditions. Cristian, this is my faculty, in my greenhouse. Growing media was actually organic soil, histosoil with over 98% of organic matter. And test cultures were lettuce, strawberry radish, and muskmelon. But the treatments were the same. In each of these trial separate, the treatments were same. Experimental design was the same, and it comprised of main factor was sodium salinity. Control without sodium salinity, then first level with 20 micromoles, 40 and 60 micromoles of sodium chloride. That was main or first factor. And another factor was actually cadmium contamination. So we have taken the soil and spiked the soil with cadmium nitrate to obtain control cadmium level as was present in soil background, then 5 and 10 milligrams of cadmium per kilo of soil. Just brief and short results. In one segment, for instance, related to cadmium accumulation in leaf tissues in those 4 crops, but you can see at different timing. For instance, in muskmelon, yeah, interesting. In some, you can see and clearly and visibly influence of salinity was visible. For instance, sodium salinity, how was influence on growth and yield of some tested plants. But I won't talk too much about the influence on growth. I will talk about cadmium accumulation. But it is interesting to see that actually in each test species, salinity actually reduced the yield, the growth, and other growth parameters. For instance, you see here with strawberry, all plants in this treatment were destroyed after first vegetation season, while these first 3 treatments, they were continued and with vegetation in next year, but actually the highest salinity level actually totally destroyed plant in strawberry, which was confirmed as the most salt sensitive among these species. Also, you can see some visible symptoms of salt stress, especially in lettuce and of course in vines of strawberry. But okay, the topic is cadmium accumulation in leaf tissue. Interesting. We got here two sampling dates. L25 means leaves were sampled 25 days after commencement of salinity treatment. L50 means that leaves were sampled 50 days after starting with salinity treatment. Similar to lettuce, 20 days after salinity treatments and 30 days after salinity treatments. What is visible? Actually, it is visible that salinity improved cadmium accumulation in leaf tissue. For instance, here by almost 90%. Here, during the second sampling, by almost 50% in comparison to control treatment. Also, positive effect of salinity on cadmium accumulation in lettuce, in strawberry, but also in radish. This is the influence of cadmium contamination. It was expected that more cadmium in soil will increase also in higher concentration of cadmium in leaves. Okay, it was logical. But interesting how much that influence was. For instance, here 15 times, 14 times, 16 times, 20 times even, yeah? Cadmium contamination, increased cadmium accumulation in lakes. Okay. But as you could saw, in muskmelon and lettuce, actually, this influence was lower over the time. During the first time, increasement was almost 90, and during the second times, almost 50%. So, how to explain that? How to explain it? Okay. Relatively easily, let's say, but what happened actually? Here you can see the cadmium concentration in drainage solution. So the plants, as you saw here, actually were grown in a pot filled with organic soil, or here in bags filled with organic soil, but actually each pot or each bag has the holes for drainage of excessive nutrient solution. So what happened over the time? Over the time happened that concentration of cadmium over the time was lowered about 2 times. So it is very important from the environmental protection, because the leached concentration of cadmium from such growing media can reach the surface waters, groundwaters, and affect the waters, for example. Okay? That's also important. What is also important? For instance, by using the one modeling approach, one free of charge chemical speciation model software, Visual Mintec, developed by Georg Peter Gustafsson, and using that by one specific model which is incorporated into the Visual Mintec. Actually, you can predict speciation of certain elements like cadmium, and we did that. We did that in certain drainage samples, 25 and 50 days after the starting with sun. And actually, this explain increasing of cadmium phytoaccumulation with sodium salinity and cadmium contamination. So what is here important? Here is important that for instance, this is actually controlled treatment. You see, sodium chloride 0, sodium chloride 20, 40, and 60 micromoles per liter, micromoles, sorry. What happened in control treatment in comparison to other treatments? For instance, let's have a look this cadmium species, free cadmium, which is the most bioavailable form of cadmium. You can see that in control treatment, that form was actually almost negligible, about 0.1%, and that's it. And actually, the most of cadmium in control was complexed with organic matter. While in other treatments, you can see decreasing trend in that form, organic form, especially in the highest sodium chloride treatments. And with salinity, I just remember you, we had the highest level of cadmium in leaves. Okay? If we want to graphically present that situation 25 days and 50 days after starting with salinity, then you clearly can see that actually, and the concentration of cadmium were also, as I said, about 2 times lower than during the first sample, but actually distribution was quite similar, 50 and 25 days. Okay. Yeah, now I will show you something interesting, also related to chemical composition, but chemical composition of one generalized rhizosphere solution, which was also modeled in the same program, in the same model, Nikodonov model. How we obtained this composition? We obtained this composition, let's say, from normal mineral soils, from the literature, and it corresponds very well to non-contaminated mineral soils. For instance, 1.8 micromoles of nitrate, 0.5 micromoles of phosphate, sulfates, and so on and so on. And here we got metals of interest that we want to model in visual mimicry with two conditions: with normal, non-contaminated, and for contaminated conditions. Contaminated are placed in brackets, and these are, let's say, for normal conditions. The same is for some organic compounds, but low molecular weight organic compounds, like citrate, malate, formate, oxalate, and so on and so on, overtaken from literature, scientific literature, which is explained here. And this is actually that we have also assumed. We have assumed concentration of high molecular weight organics, fulvic acids, and of course dissolved organic carbon, which we were set as 10 milligrams per liter. 10 milligrams per liter is, let's say, concentration of DOC in normal mineral soils. And we did model for wide range of pH from 3.5 to 9. So let's have a look what we have obtained for 3 of elements, only for 3 of elements. Yeah, here you can see what happened with cadmium speciation in that modeled rhizosphere solution. Left bar means uncontaminated, and right bars means contaminated conditions. But you can see actually they are quite similar. They are quite similar in contaminated or non-contaminated, but difference is actually starting here. You can see what is happening with the most bioavailable form of cadmium. It has decreasing tendency from 3.5 to 9 pH range, and of course opposite trends with inorganic complexation, let's say from here up to 9. Similar, but also you can see activities or concentration of certain cadmium varieties. For instance, free cadmium form, cadmium chlorocomplexes, and cadmium malate, cadmium complex to fulvic acid, and cadmium complex to carbonates. You can see there how their activities are changed over the pH range for cadmium, but also look at for zinc, quite similar speciation to zinc. We will see later on when we put these two situations together. So zinc speciation as cadmium showed strong potential for inorganic complexation, inorganics, chlorides, carbonates. When? From here, when pH rises, when pH rises, and especially in relatively low presence of organic matter soils. So it can explain why is zinc the most common deficient crop micronutrient under higher pH in the world, alkaline soils, because it's mostly complex to carbonates, mostly, and it's not such available as in its other forms, especially as it's in free form. And interestingly, it is known that zinc and cadmium has very similar chemical speciation, let's say, under the same environmental conditions. You can see very similar with activities of cadmium and zinc, and also in distribution of particular species of zinc and cadmium under the wide range of pH, especially in this range, they are both mostly complex to carbonate forms. But opposite to cadmium, opposite to zinc, you can see the chemical speciation of copper. It is known actually that copper possesses one of the highest affinity to soil organic matter constitutes among all trace elements. And actually you will find here almost all copper in all ranges, in all this range, was present at low concentration as free copper. It was mostly complex with organic, you can see, organocomplexation over carboxylate anions dominated actually in this pH range from 3.2 to let's say 6, and thereafter 6, organocomplexation continued increasing with pH rising. But the point is that actually low portion of copper as free copper was present in this model situation. So it is mostly complex with organics or with other, like is in this case fulvic acid, or is oxalate or malate, and fulvic acid is a part of high molecular weight organics. It is interesting. And finally, some conclusions from this my presentation. We can say that soil organic matter has, as a pedosphere multiplex, has actually irrepressible ecological roles. Today we didn't spoke about its role in global cycling, but it has very important role in that segment, also in carbon and nitrogen cycling. Also, you have seen that actually SOM is of vital importance for organic and inorganic contaminants because of their absorption and consequently influencing on their mobility. Unfortunately, intake of most toxic elements and essential elements is conducting over food, either vegetable, either fruits consumption, and it's actually the main route of many trace elements into our body, food consumption. Trace elements complexation with organics ligands like humics, carboxylides, and other organic ligands, or inorganic ligands, principally chlorides, can enhance desorption from soil solids and thus mobilize trace elements in dissolved phase, improve their root uptake and accumulation, although complete mechanisms are still not in fully clear. Also, we have seen that addition of humics, fulvic acids, humic acids can impose contrasting effects to trace elements. From my example, from our example, you could see that actually influence was suppressed uptake of certain metals, but on other case, in other studies, they obtained totally opposite results. But it means that interaction of metals with the solid phase of humics may lead to their immobilization and thus decrease their potential negative environmental influence. Also, with reddish, you saw that actually humic acids decrease phytouptake of certain metals, but without negative influence on the certain test plants. So we can conclude that actually humic acid controls the mobile free cationic forms of trace metal elements and their uptake by crops. Increased uptake and phytoaccumulation of cadmium, zinc, and copper in the presence of excessive inorganics like chlorides in the soil rhizosphere have confirmed in a wide range of test species, suggesting that metallochlorocomplexation could also be one of the main mechanisms for enhancing metal uptake and metal phytoaccumulation, according to certain person. However, similar but contrary influence and relationship between copper, zinc, and cadmium levels in soil solution and their shoot phytoaccumulation under salinity has been recently confirmed in wheat, but also, as you can see, in radish. What we can explain mainly by zinc to cadmium competitions, because you saw they are they got quite similar speciation in the same environmental conditions. So because of competition among those elements, that's how we can explain this influence. Namely, in the complex soil matrix, like is for instance presented here graphically, actually cadmium and zinc and other metal elements interacts with different soils constitutes, with clays, with hydroxides. Also, they can precipitate and became unavailable to plants. But the very small portion of free metal forms, like this cadmium or zinc, is actually present in the nutrient solution at relatively small concentration. So, at the rhizosphere versus bog soil, due to pronounced rhizodeposition and microbial secretion that Alex very well knows, because of secretion of different inorganic compounds, the protonation and release of organics and so on, may be expected a favorable ratio of relatively mobile free trace metal elements, for instance, zinc and mangan, and or their simple complex form, for instance, cadmium low molecular weight organics, cadmium chlorides, zinc chlorides, and so on and so on. Therefore, finally, may be concluded that solubility, mobility, or bioavailability or toxicity of trace elements are markedly different on the relatively small spatial resolution in bulk soil in comparison to rhizospheric soil. And finally, I would just like to say that I'm very glad that I'm part of this project, Fondecyt project, leading by Dr. Cristian Gergichevich. And because of that project, I'm actually here and giving my talk today and two other talks next week. Thank you very much for your attention, and I expect some for discussion. Yes, yeah. Please. Yeah, thank you.
Thank you, Gabriel. We have a short time for some questions or commentaries, suggestions. Someone?
Don't be afraid. Don't be ashamed.
Don't be ashamed. Or in Spanish, if
yeah, of course.
In Spanish, if you want. Gabriel, in relation to the cadmium pollution, in central Chile, we have recently a problem with cadmium accumulation in corn crops, in tissues. But this cadmium contamination or toxicity in tissues, many people think that is due to the involuntary addition, because many pesticides include cadmium in the formulation. And under your experience, you note that similar condition in crops in Europe?
Yeah, thank you for the question. Actually, it is known that one of the main roots of cadmium, for instance, to agricultural soils are occurring under the fertilization. Some of, for instance, raw materials that are using for productions of, for instance, phosphates and other chemical nutrients can be overloaded by excessive concentration of cadmium and some other elements. So quality of, let's say, fertilizers, quality of raw materials for phosphates and so on can influence the soil quality in agricultural ecosystems. That's one thing. Another thing is that also you can introduce, let's say, trace elements, cadmium and so on, with irrigation, with water used for irrigation. If water quality is not optimal, if it's, I don't know, for some industry that can be overloaded by not only metals, with other organic contaminants, it is also one of possible way to introduce metals like cadmium and inorganics to your agricultural soils. But yeah, that's actually two principal routes of let's say, contamination of agricultural soils by trace metal elements.
Yeah.
There are in some other
Uh-huh.
aerodeposition and so on. Erosion, okay, as you saw, and fluid deposition, as you saw maybe on my, okay, we don't have proof for that. But if we don't have industry in that region, from where is that lead or zinc?
Yeah.
Must be from upstream, which over the time was deposit, and it was confirmed in many studies that most of metals actually can be deposited by fluid deposition over the time in certain amounts. And I believe that it is also the case in those two examples.
A similar situation occurred with copper.
With copper, copper, yeah.
Usually during winter, autumn, and winter time, during latency period of many fruits, the application of copper is usual management. So avoid,
of course,
to avoid the disease against pathogens,
in Croatia
pathogens like bacteria or fungi. So the real impact of copper pollution after this agronomic management is unknown. Still have a little study.
My colleagues from department in really nice studies, published studies, have confirmed what you said now. Actually, copper application of copper sulfate is very, very common in our vineyard management. At the beginning of vegetation, first application of copper as copper sulfate is one of treatment as a normal, let's say, intervention for prevention of some diseases and bacteria in vineyards, and also after or during the vegetation there are also one or two intervention for pathogen prevention in vineyard. And really they have confirmed hotspots in vineyard region with copper, especially in the topsoil. It is known that copper is, as was shown here, copper is very, very adsorbed to soil particles and has low ability to be leached or transported by water to deeper soil profile. So it was confirmed in many vineyard region in Croatia, but also in other regions. But copper is very interesting for that point of.
Someone has a question?
I have. Thank you for your presentation. I was thinking about your results of humic acids.
Okay.
And they were contrasting with other results. And what do you think about that? Because it was the nature of the humic acid, maybe. Of course, there were different experiments, but what do you think about what they were different?
Okay. Those two studies were conducted in soil. In soil, and our was conducted actually in a nutrient solution.
So you have isolated and you have absolute control of the humic acid that you use in the experiment, and the other experiments not.
For instance, here in this example here, they got 3 treatments with humic acids: 0%, 1%, and 2%. And it's clearly visible that each treatment with humic acids increase. So, look, on this slide here, one of very interesting question, but one of possible explanation can lie here. This is let's say soil environment. So they have applied the humic acid in the soil, but that some element can be taken up by plants, it has to be present in dissolved phase. So what can be the reason for that? For instance, in soil, humic acid can help that copper, for instance, or cadmium, in this case cadmium, can be replaced from the solid soil phase and be released to dissolved soil phase. And by organocomplexation with humic acids, it can be it can reach the roots of plants quickly, more quickly, and be taken up as fragments of organics, like small organic fragments of humic acids and metals. That's one explanation. So in soil, if you got lots of clay, it is also possible competitive sorbent for cadmium, like is humic acid also. So in soil, probably that complexation with humic acid improved that cadmium be maybe released from solid soil phase, be complexed with soil organics and transported to roots of tobacco more quickly than it would be in the case of nutrient solutions only. Okay, because our study was conducted in nutrient solution media, in totally different media than soil, which is much more complex. And on complexation of cadmium, you got different influence from different soil particles. Yeah, that's one possibility. And I remember that in this paper, Evangelou and associate, they also assume that one possibility for such influence could be that cadmium was complexed to humic acids, and smaller segments of cadmium organocomplex could be also taken up by tobacco roots. Okay, but yeah, you have to consider experimental conditions, soil and nutrient solution. This is a much more simple matrix than in soil. Okay. Yeah, I don't know. Did you understand me? But
yeah.
So in soil, let's say organics can accelerate the mobilization of cadmium, of trace elements, to the roots.
Yeah,
okay?
Because I was thinking only in the nature of the humic acid maybe was different composition, because it's a matrix of things.
That's right. This was soil, and in our it was distilled water with chemicals. Okay. Yeah, and in soil, you have everything, and clays and everything. Yeah, matrix is totally different. Yeah, but also I remember this paper. I remember very well this paper because why? We have recently published this paper, 2018, and one of reference was also this one. So I remember this. they were also discussed as possibility that small fragments of humic acid could also enter the plant root as cadmium organocomplex. They assumed, okay, but anyhow, in soil, definitely complexation of cadmium with humic acids highly likely accelerated transfer of cadmium to tobacco roots. Yeah.
Question? Well, with Daniela Figueroa, she's an agronomy student, undergraduate. With she, we work in objective of this Fondecyt project related to the amount of oil in hazelnut fruits. Yeah, I told you. So we preliminary results. We note that zinc has an effect on the world. Has an effect on level of oil in fruits. So when we apply zinc, foliar,
then you obtain more.
We obtain more oil content in fruit, but still not now about which is the oil profile. But we know that the zinc oil content is increased after zinc application.
Is that good for the quality of hazelnut? Or?
Yeah, the quality of fruits is evaluated by oil content. Is one parameter.
Yeah, yeah.
So that's the problem is that zinc in leaves is very high.
Which is
800 ppm in leaves.
Okay.
With David in the first season, evaluated season, we detect higher. Higher zinc level concentration in leaves than reported for specific.
Have you detected maybe some kind of toxicity?
Yeah,
under the
and in fruits the content is approximately 70% of leaves content. So in the leaf 70%. So our preoccupation is in that zinc content in fruit could be dangerous maybe.
That's right.
For human consumption.
That's right.
So, but oils are increasing, but the zinc content in fruit is very, very high.
Interesting. Yeah.
Okay, more questions, commentaries? No. So,
guys, thank you very much for your attention.
On behalf of BioRen and my Fondecyt project, I would like, we are grateful for your interesting introduction. So about your research line, so the mix of mix different topics.
This topic and this presentation is probably the most, let's say, scientific, difficult maybe to follow. Another two will be, let's say, more relaxed, more general, let's say, and especially the last one will be even more practical, how to deal with some water management in practice. This you can't talk about trace elements and be simple. I mean, I can't. But maybe it was too difficult to follow. But sorry, I'm sorry.
Also, I would like invite to PhD students and some researchers here, like Dr. Luego, to your next talks on Monday. Monday 6.
Of course.
And here in the same place at 10 a.m.
Topic will be with water management. Yeah, in agri ecosystems.
Okay. Thank you, Gabriel.
Thank you.
Thank you, guys.